So what is enzyme inhibitor? Inhibitor are the substances which alter the catalytic action of enzymes and consequently slow down or diminish the enzymatic reaction. The process is called enzyme inhibition. An enzyme inhibitor prevents the formation of either enzyme substrate complex, or it can prevent the breakdown of enzyme substrate complex to enzyme and product. Inhibition is very important for the development of the area of research in the field of biochemistry and ecology. So how inhibitor works? In case of enzymatic reaction, general enzymatic reaction which occur as enzyme first bind to substrate at the active site to form enzyme substrate complex, and this enzyme substrate complex finally degrade to enzyme and product. But in case of inhibitor bind to either free enzyme or enzyme substrate complex and prevent for the formation of product. Enzyme inhibition are two types: reversible and irreversible enzyme inhibition. Reversible enzyme inhibition bind to the enzyme through non-covalent bond, but irreversible enzyme inhibitor bind to enzyme by covalent, which is a strong bond. Irreversible, there are different type of irreversible enzyme inhibition. Some example are here. In case of phosphorylation, where the phosphate group is attached to the tyrosine, serine, threonine or histidine residue present on the enzyme and form the phosphorylated form of enzyme. Because of the attachment of phosphate group, the conformation of enzyme is changed, so their activity is also affected. In case of adenylation, EMP molecule is bind to the enzyme at tyrosine residue and it's modified. In case of uridylation, UMP molecule is attached to the tyrosine residue of enzymes and its structure is changed. In case of ADP ribosylation, ADP ribosyl group is attached to the arginine, glutamine, cysteine residue and modified it. In case of methylation, methyl group donor is an S-adenosyl methionine, which donate methyl group to the glutamic acid residue on the surface of enzyme. So enzyme are methylated and their activity is also affected. There are three types of reversible enzyme inhibition. First is competitive inhibition, second is non-competitive inhibition, third is uncompetitive inhibition. In case of competitive inhibition, the inhibitor molecule is structurally similar to the substrate. So inhibitor bind to the same site where normally substrate is bind, that is active site. So after binding of inhibitor, substrate are unable to bind it in that site, and because the presence of inhibitor, the affinity of substrate is affected with enzymes. So Km in case of competitive inhibition, Km is increased while no change in Vmax. When we draw a plot between 1 by S and 1 by V, we found a same intercept on 1 by V0 axis where we obtain 1 by Vmax, and different type of intercept are obtained at X axis, indicate that difference in their Km value. The best example of competitive inhibitor is the enzyme succinate dehydrogenase, substrate succinate, and its competitive inhibitor malonate. Structure of malonate is just similar to succinate, so malonate compete to the binding of active site with succinate. If the succinate is bind to the active site of succinate dehydrogenase, then reaction is occur. If malonate bind at the active site of succinate dehydrogenase, then reaction is diminished or prevented. In case of uncompetitive inhibition, uncompetitive inhibitor bind to the enzyme substrate complex only and inhibit the reaction. This type of inhibition is also called anti-competitive enzyme inhibition. In case of uncompetitive inhibition, Km and Vmax both value are decreased. We found different intercept on 1 by V and 1 by S, indicate that in the presence of uncompetitive inhibition, Km and Vmax value is changed. In case of non-competitive inhibition, which is also called mixed type of inhibition, inhibitor is bind either free enzyme or enzyme substrate complex and inhibit the reaction. In this type of enzyme inhibition, Km value is unaffected while Vmax is decreased. What is the significance of enzyme inhibition? The most important significance of enzyme inhibition is the development of new drug for the treatment of certain type of pathogenic infections, diseases, and take care of mankind's. Enzyme inhibitors also occur naturally and are involved in the regulation of many metabolic pathways. Regulation is very important step in metabolism that regulate any metabolic pathway, means it metabolic pathway is required, then the reaction is going on. If the metabolic pathways products are not required, then these metabolic pathways are inhibited by regulatory enzymes, and I will discuss regulatory enzymes in next of my lecture.